Okay, so now we're moving into cells, and so just to reorient yourself, as a reminder, we started off with atoms, which are the smallest stable unit of matter. We put groups of atoms together to get molecules, small and large. And at the end of our last lecture, we ended with the 4 types of macromolecules, the 4 types of large molecules that are found in cells. So remember, we group molecules together and we end up with a cell. Okay, and as a reminder as well, cells are the first things that are living. Carbon, an atom, not alive, but cells are living. And in fact, everything we have discovered so far that is alive is made up of cells. Okay, and so that's what we will go into next. So often what I have my students do is I tell them to look at this picture and or look at their neighbor's face, and they don't have to get quite this close to their neighbors, but to tell me what they see. And usually they'll list a lot of different features, but eventually what we come to is that what you're seeing when you look at someone else is cells. Either a cell or something made by a cell. And so that's what we'll go into. Now cells generally are going to have 3 major parts. And so here's our cell. And if you're following along on the notes, then they would be right here. So these are the 3 major parts of the cell. Okay, so plasma membrane. We know that that is this boundary. That's what separates outside from inside. Okay. We then have the nucleus. And so remember, we ran into the word nucleus before with an atom, and it was the center of the atom, the thing inside. And so if you look at a cell, well, here's that thing in the center of the cell. Now, in reality, sometimes the nucleus may be squished off to the side, but it got its name based on being a circle inside of a cell. Okay. Now, everything between the nucleus and the plasma membrane, so everything between nucleus and plasma membrane is called cytoplasm. Cytoplasm. And we could see it in 3 dimensions all around. Okay. Now the cytoplasm is going to be filled with watery like liquid. And then also these larger structures. And these larger structures are called organelles. Okay. So just to follow along with the notes, plasma membrane is the boundary of the cell. Nucleus is that big circular structure in the middle of the cell. It contains DNA. The cytoplasm is going to be between the nucleus and the plasma membrane. The cytosol is going to be the watery liquid of the cytoplasm. And then the organelles are going to be the structures with specialized functions in the cell. and sometimes they're called subcellular organelles. So organelles, subcellular organelles, it's the same. Remember, sub means below, so it's something that's smaller than the cell. Okay. Okay, now the rest of this homework lesson, what it does is it takes you through one way of looking about the organelles. And one thing that you can do as you study these, I would recommend that you turn to page 66 and 67 in your textbook, or page 42 and 43 in your lab manual, and that you look at those as you learn these organelles. Okay. Also, you'll find a paragraph in your textbook that talks about membranous versus non-membranous organelles. That means, is it surrounded by its own membrane? Okay, so if we look back at this picture, is this little structure, does it have a phospholipid bilayer wrapped around it? Or is it just a large group of proteins? Here, does this have a membrane? Is it phospholipids? Okay. So that is what this is asking you for on this page of the homework. And then what the rest of the homework does is it looks at what do cells need, and then what are the things that help them accomplish that need that they have. So if you're following along on your notes, I'll just give you these needs, and then I would suggest you go through and to learn them. I'm going to give you several different ways to learn these, and one way to learn them is to go through and see which organelles help accomplish this function. And you can have more than one organelle listed here, and organelles may fit in several different functions. Okay. So one need of the cell is to form lipids and make functional proteins. So what are the things that help a cell do that? Another thing they do once they make the lipids and proteins is they have to transport lipids and proteins to the right location in the cell. Okay. Another thing that they have to do is they have to rid the cell of harmful chemicals. Our environments have all sorts of chemical assault. Some of these reactions that happen inside of a cell produce harmful chemicals. So they have to rid the cell of those harmful chemicals and recycle old molecules. A lot of times molecules can be damaged, and so they're not going to function as well because their structures have changed. And so how do we recycle them? Tear them down and then reuse some of the parts. Another thing that cells need is they need a certain shape. Remember, structure, function. They also have to have physical support. Okay, so you don't want them just like a little blob. If you think about our bodies, how do they get that physical support? How are we not a blob on the ground? Okay, and then something else that our cells have to do, some of them, is they have to have movement. So what are the things that allow them to move? And then a final need of the cell, big time, is going to be energy. So my suggestion of how to use these notes, and don't memorize the needs of the cells. These are kind of arbitrary that I came up with, but I want you to think about what are the organelles that actually help these things to occur, and that's one way of learning these organelles. Another way of learning the organelles is going to be a learning activity that I'm going to show you. Okay, so this is showing us the printout from a file that is attached in your Blackboard website, and it's showing you some of the different parts of the cell. So here we have cartoon parts, and I tried to make them to somewhat match the images you have in your textbooks and in your lab manual. Okay, and here are some functions. And then here are the names of the parts. So what I do with students in my class is I have them actually go through and construct a cell, because often this is a little bit, helps the information stick a little bit better than if I just go through and talk about it. So what I have students do first is you have to think about, okay, what are the three major parts of the cell that we talked about? Well, cytoplasm, nucleus, and plasma membrane. So do you see anything here that looks like a plasma membrane? No, probably not. And so what I would do is have a piece of paper, and the first thing you can do is you can actually draw your boundary. That's the boundary to your cell, the plasma membrane. And then you can take your piece of your label that says plasma membrane and put it right on there. So we've constructed the first part of the cell. Okay, and let's put some of this aside until we're ready to build our cell. Okay, and so one of the first things that you can put is, you know, there needs to be a nucleus. Okay, and so this is the thing that looks most circular and round, and so that is going to be our nucleus. And so we can put it there and we can label it nucleus. Now, at this point, we could ask ourselves, well, what's in the nucleus? And we can use our charts, we can use the different things in our book, and we can also go through the functions that are a part of this activity, that are part of those files. Now, those functions aren't written using the exact same language as in your textbook, and that's on purpose, because I really want to make sure, do you actually understand these functions? Okay, and so if you can match them, that's great. And so my suggestion is, or actually after you look through it, then you can see there's one that says contains DNA, the genetic or heritable material. Well, that sure sounds an awful lot like the nucleus to me. And so now I've got my first subcellular organelle with its name and its function. Now, many times I have students that have asked me in class, they say, well, why do you have two of these? Well, there's a reason I have two of them. And a lot of students will recognize that these here are endoplasmic reticulum. Okay, and if you look in your book, you say, well, I've got two types of endoplasmic reticulum. I've got smooth endoplasmic reticulum, and I've got rough endoplasmic reticulum. But what makes them different? And a big hint that I'm going to give you is that with rough, ribosomes are attached to them, not with smooth. Okay, why does that matter? Let's think through this. It turns out that ribosomes attach to the rough ER because ribosomes make protein. And so rough ER is going to help make protein because ribosomes are attached to it. What types of proteins? Membrane proteins. Okay. Proteins that are either embedded in the membrane or secreted proteins. And we could even write that over here to remind ourselves, membrane or secreted proteins. Are made here. Okay? And we could put a little here. Okay? So ribosomes make protein, and if they're attached to the rough ER, then they're going to be making secreted proteins or membrane proteins. Now, are all of our proteins secreted or membrane? Well, no, some of them want to remain inside of the cell. And so we still have ribosomes there, and they're still going to be making proteins, but these are free-floating in the cytosol, okay? And these are going to make proteins that stay in that cytosol. Okay, so then smooth endoplasmic reticulum, it helps detoxify things. It's going to help make lipids. Those are two of its major functions. We've got the Golgi, which is this thing, also called the Golgi complex, the Golgi apparatus. Okay. And this structure is going to add carbohydrates to a protein. So proteins are going to travel from the rough ER. Once they're made, they travel to the Golgi. And once they're in the Golgi adds carbohydrates to it. Now, you tell me, here's our protein. We've seen this from the last lecture. And we have decided we're going to add carbohydrate to it. What have we just done to the protein's structure? We've changed it. And structure always equals function. So, what the Golgi does by adding carbohydrates to proteins is that usually it makes those proteins functional. So some proteins, they can be made here, but they don't function until a carbohydrate is added to them here. And then it turns out those proteins can be delivered to the plasma membrane. And that's a big place that they're delivered. Okay. I'm going to move this guy at free floats. It just floated up here to give me a little bit. And we should put a label on there. That's a ribosome. Okay. Now, let's look at some of these other words. So we have this thing that it looks a little bit like a football on the outside, but it's got this wavy interior. Okay. That, if you look at the pictures in your lab manual, hopefully you'll recognize it's the mitochondria. Okay. And mitochondria, some of you may have learned in high school, if you had high school biology, they often say mitochondria is the powerhouse of the cell. And that's okay to know that for high school, but that's not specific enough, because what does that actually mean? Okay, so at the college level, we need to have a little bit more information about what that is. And the big thing that I want you to know is that the mitochondria make most of the ATP, which, as a reminder, is the energy molecule of the cell. Mitochondria are going to make most of the ATP for the cell. Okay. So that's what it does. All right. Some other things. You've got two little circles. They're hard to tell apart, and that's okay. One of them is what's called a peroxisome. And this structure is going to be important for taking toxic chemicals such as hydrogen peroxide. Remember peroxisome, peroxisome, it's going to take hydrogen peroxide, which is very reactive. If hydrogen peroxide is inside of your cell, hydrogen peroxide is inside of your cell, then you can expect that your cell is going to have a lot of its molecules damaged, because that's what hydrogen peroxide does. If you've ever put hydrogen peroxide on a wound, you've probably seen it bubble up. Well, that's what's going on with hydrogen peroxide inside of a cell as well. It's just going in there and bubbling up and tearing apart the molecules. So peroxisomes take care of it, and other chemicals that are similarly reactive. Okay, this other circle here is what's called a lysosome. And basically, the lysosome is a big recycling factory. It's going to take those polymers and anything that comes in there, it's going to break it up into monomers, and then these monomers can be used, recycled, and used in other places in the cell. So, a lot of times what happens is the cell will actually scoop up stuff outside of it and take things from the plasma membrane to the lysosome, where it'll chew it up. Okay. All right. So these are some of the different subcellular organelles, as well as their functions and names. Now there's one part that I want to point out. You notice that we've got proteins flowing from rough ER, or rough endoplasmic reticulum, to the Golgi, to the plasma membrane, and sometimes things go plasma membrane back to lysosome. These arrows, where things move, are called membrane flow. Okay? And so, just to be clear on that, membrane flow, membrane flow, membrane flow. Okay? The reason why it's called membrane flow is because to deliver these proteins, you also have to deliver lipids. So lipids and proteins are flowing from one location to the other. Now, a couple of other parts of the cell that are a part of this activity is you have this word, extracellular matrix. And let's break down this word for just a minute. So if we break down this word, extracellular matrix, extra, in this case, is going to mean outside. And cellular means cell, so literally it means the stuff outside of the cell. Now, this is important because where does that stuff come from? Well, it turns out that cells are going to be making things inside of them. Okay? They make all sorts of materials, and they spit it out. And so some of those materials are going to be outside the cell. Okay? So, all of this squiggly line here is extracellular matrix. I sometimes tell my students, and this is a little gross, but they seem to remember it, that extracellular matrix is essentially cell vomit. The cell makes it, it comes from inside the cell, it vomits it out. We'll talk in a minute why it's important. Okay, now we also have this word here, cell junctions. So I think about the word junction. If you're driving and you see the word junction, that means that two roads have come together. So a cell junction means a place where two cells have come together. So if I was going to draw a cell junction in, I draw another cell. Okay, so here's the plasma membrane of my other cell. And a cell junction is generally a set of proteins that bind them together. Now the last one we haven't talked about is cytoskeleton, and I'll talk more about that in a minute. Okay, here I'd like to talk for a minute more about membrane flow and to show you exactly what's happening. So this is a video that's embedded in your PowerPoint feature, and here we've got our nucleus, our rough ER, our Golgi, our plasma membrane. And what we're doing is we're taking a close-up view of first the ER, okay? So here we have a protein that's been made inside of that ER, and we're having membrane flow to take it from one location to another. Now what just happened there? This little thing, it's a pinch off of the ER, is called a vesicle. And inside it, it's got that protein. And you notice it's going from the rough ER to the Golgi. And look what just happened. That vesicle fused with the Golgi. Keep in mind that the reason that can happen is because rough ER is bounded by a membrane, Golgi is bounded by a membrane, and vesicles are just a little pinch off of the membrane. That protein goes through the Golgi, it gets carbohydrates added to it, becomes functional, and now we're going to have another vesicle pinch off, and this is our vesicle, we've got our protein inside. It's headed to the plasma membrane. Now, this is showing the plasma membrane. You should recognize those phospholipids with the head and the tails. Okay. So let's get a close-up view, and here's our vesicle. Also, phospholipids surround it, and inside are the things that the cell wants to secrete. This is the extracellular matrix being made. This is the cell vomit inside of here. So we come up and then look how those phospholipid layers fuse and then dump out the contents. So that's how membrane flow works, and that's how extracellular matrix is made. This is all going to become part of the extracellular matrix. This can also go backwards. Remember I said this plasma membrane, and look, it's making a vesicle the other way. And it's taken some contents, bringing it inside the cell, and that little vesicle may be headed straight to the lysosome. Okay, now the question is, if we're looking here at membrane flow, and we want to say, well, how does that vesicle know to travel from here to here? Why doesn't it go from here to over there? Well, this is where part of the cytoskeleton comes in. Okay, there's three types of cytoskeletal elements, and let me list them for you. So we're going to have microtubules, microfilaments, and intermediate filaments. Okay, let's talk about microtubules first. So it turns out that in the cell there's a highway system, and all along here are going to be these microtubule tracts. And they're going to help as a highway system those vesicles jump on and know exactly where to go. Now let me show you a video that's pretty amazing. Okay, so basically they took a cell and they mushed it up, and all of these little circular things here are vesicles. This right here is a microtubule, so you can see it's like a little track. And let's watch what happens. Okay, so vesicle or an organelle, and watch what happens. Okay, this is for real. See how that thing jumps on and they travel down. That is the highway system of the cell, and it helps things get from one location to another. Notice it's a two-way highway. It can go either way. But these highway systems are all throughout the cells. Vesicles jump on and get to their destinations. Okay. Now, I want to make another point here. We were talking about extracellular matrix, and let's just remind ourselves extracellular matrix is going to be all of this stuff over here, okay, outside the cell. And so why does it matter? Okay, well, here what we've done, what we're looking at here is muscle tissue. And if you notice the red here, that's the muscle, but we have all this other stuff on the outside. Okay, so red here is muscle cells. And then look at all this spider web sticky stuff that's holding those cells together. That is all extracellular matrix. It's outside the cells. It sticks the cells together, but importantly, it is made by a cell and the cell secretes it. The cell vomits it out. Okay, so extracellular matrix. And so in this case, that matrix basically packs all those cells of the muscle together. If you didn't have that, these cells would just be wiggling all over the place, and it would be very difficult to have a coordinated muscle contraction. Okay, now this here is actually showing you a cell. So we've gone really close up with the microscope, and what I hope that you can see here is this right here is the boundary to the cell. Okay, so the red is pointing out where it's inside the cell. Now the red is actually staining these microfilaments. Okay, so we can stain them and look at them under a microscope. And you see they're all over the place, but they're particularly strong on the edges of the cell. Okay. Now it turns out microfilaments, they're pushing out on the plasma membrane, and they can change the shape of the cell. And you can actually see this. Here's an amoeba. It's a one-celled organism. Here's the outside of the cell. And this is all microfilaments pushing on that plasma membrane. And look what happens to that amoeba. It keeps changing the shape of the cell, and that's actually what gives this cell movement. Okay, so pretty amazing stuff. Finally, we've got intermediate filaments. And again, let me point out the boundary to the cell. So the boundary is going to be here. Okay. Now, you notice you do see intermediate filaments everywhere, but unlike the actin where it was super strong on the edges, it's there. But actually some of the stronger places are here. Now this is in the center of the cell. It's circular. What would you guess this is? You said nucleus, you're correct. And so these intermediate filaments are really mostly there to give structure and support to the cell, so that if something presses on this cell, presses down on it, there's a little bit of structure there to resist it just being totally squished. Okay. And so that's what these intermediate filaments are, is they really give structure and support to the cell. Okay, and last, what I'd like for you to do is that both in your notes and also in your homework activities, there are a number of activities that help you practice your knowledge about these subcellular organelles. Okay, so here is a chart, and you could fill out the function. Is it bounded by membrane, thus membranous, or not, non-membranous? And is it a part of the membrane flow process? And so these are here to once again help you practice your knowledge. You can practice drawing an animal cell yourself. You can practice drawing membrane flow. You can use the information we just talked about to help you learn about these 3 cytoskeletal elements, where they are, what they do inside the cell. And then lastly, there's a thing for you to help to think through what's the normal function of an organelle, and then what function, or what would be the effects if this organelle didn't function properly. And just to kind of give you a quick sneak peek, this individual has Gaucher disease, it has defective lysosomes, and just to let you know, this kid is not malnourished. He does not, he's not super fat. That is his liver poking out. It is so large that it is poking out like that. Okay. This is another one, a defective Golgi apparatus. This individual ends up with Tangier disease. You can read about that. And Pearson syndrome is a defective mitochondria. So again, I'm not necessarily asking that you memorize these diseases, but I want you to think through the logic of it. What does it look like when these organelles aren't working properly? I also want to point out that microtubules can also be involved in cilia and flagella, and so here's some videos that you should watch in addition about microtubules. Okay, so this is cells, and I hope that that gives you some tools, both with the at-home learning exercises. You could use these over and over again until you really have got them down, with the notes packets where you can go through and fill in this information and fill it in again. Maybe fill it in the first time and then keep doing it until you can get it by memory and you've got all of this memorized. Okay. And then also with the homework activities as another place that you can practice reading about and learning more about these organelles